Hello and welcome to some biology class. Friends, today we shall discuss about CRISPR-Cas9. CRISPR is an acronym for Clustered Regularly Interspaced Short Palindromic Repeats. It is a family of DNA sequences found in the genome of bacteria and the archaea. The sequences are derived from the DNA fragments of bacteriophages which had previously infected the prokaryote. They detect and destroy the DNA from similar bacteriophages during subsequent infections. Hence, the sequences play a key role in the antiviral defense system of the prokaryote and provide a form of acquired immunity. The other component of the CRISPR system is CRISPR-associated protein 9 or Cas9. Cas9 is an enzyme that uses CRISPR sequences as a guide to recognize and open up specific strands of DNA. Cas9 together with the CRISPR sequence form the basis of a technology known as CRISPR-Cas9, which is used for genome editing within the organism. For developing CRISPR-Cas9, Emmanuel Carpentier and Jennifer Doudna was awarded Nobel Prize in 2020. So, the CRISPR-Cas9 system consists of two components. One is Cas9 and the other component is the CRISPR sequence, which together are used for genome editing within the organism. The genetic information that produces the CRISPR-Cas system is found at a location in the genome of the bacteria and the archaea. The location, which is known as the CRISPR locus, consists of two different parts. One part is a small cluster of genes encoding Cas proteins which play a specific role in the CRISPR system. The other part is the CRISPR array, which is made up of an 80-rich leader sequence followed by short repeats separated by unique spaces. The size of the CRISPR repeat varies from 28 to 37 base pair. The CRISPR-Cas system can be divided into two main classes, class 1 and class 2. Class 1 system is further subdivided into type 1, type 3 and type 4, whereas class 2 CRISPR system is divided into type 2, type 5 and type 6. The CRISPR system has a role in adaptive immunity in bacteria. CRISPR-Cas mediated immunity has different stages. Stage 1 is acquisition of species, which is mediated by the proteins Cas1 and Cas2. In this stage, sequences derived from DNA fragments of bacteriophages which had previously infected the bacteria are inserted into the CRISPR locus next to the leader sequence. These are the unique spaced sequences which are derived from the DNA fragments of bacteriophages which had previously infected the bacteria. Stage 2 is synthesis of a long primary CRISPR transcript. This is the primary CRISPR transcript which is synthesized from the CRISPR array. The mechanism to produce CRISPR array differ among CRISPR-Cas system. In type 1e and type 1f system, proteins such as CA6E and CA6F respectively recognizes the stem and loop created by pairing of the identical repeats that flank the CRISPR RNA. These proteins cleave the long primary CRISPR transcript at the edge of the paired region, thereby leaving a single CRISPR RNA along with a small remnant of the paired repeat region. So, in type 1E and type 1F CRISPR system, these repeats are identical, because of which base pairing occur between them that results in the formation of stem and loop. In type 3 CRISPR systems, CA6 proteins are also required, but the repeats do not form stem and loop. In case of type 2 CRISPR systems, CA6 proteins are not required. Instead, the primary CRISPR transcript is cleaved by ribonuclease 3 to form the CRISPR RNA. Functional type 2 CRISPR-Cas system require an additional RNA known as transactivating RNA, which is complementary to the repeat sequence present in the primary CRISPR transcript. As a result, base pairing occurs between the transactivating RNA and the repeat sequence that results in the formation of a double-stranded RNA, which is then cleaved by the enzyme ribonuclease 3 to yield CRISPR RNA. Unlike the other two types of CRISPR systems, in this case, the CRISPR RNA do not contain the full spaces, so the spacer is truncated at one end. In case of type 2 CRISPR system, the primary CRISPR transcript is cleaved by the enzyme ribonuclease 3 to form the mature CRISPR RNA, who is then associated with Cas9 nuclease to form the interference complex. These interference complexes can detect and destroy the DNA fragments of invading bacteriophages if they are similar to the phages which had previously infected the bacteria. The CRISPR-Cas9 system, which has a role in adaptive immunity in bacteria, is the most recent addition to the genome editing toolbox. Type 2 CRISPR system protect the bacteria against the DNA from invading viruses and plasmids via RNA-guided DNA cleavage by Cas proteins. As the target site recognition by Cas9 endonuclease only requires a seed sequence within the CRISPR RNA and a conserved protospacer adjacent motif, CRISPR-Cas9 system has emerged as the most flexible and user-friendly platform for genome editing. The CRISPR-Cas9 system, which is used for genome editing, has been simplified. It consists of a Cas9 restriction endonuclease and a guide RNA. The guide RNA consists of CRISPR RNA and a trans-CRISPR RNA. The CRISPR RNA guides the Cas9 endonuclease to the target site where this enzyme introduces a double-stranded break in the target DNA, thereby enabling genome editing. Cas9 is a type 5 restriction endonuclease. In this figure, this is the target DNA. And this is Cas9 restriction endonuclease, which along with the guide RNA form the CRISPR-Cas9 system. Cas9 is the type 5 restriction endonuclease which is guided by the guide RNA to introduce a double-stranded break in the target DNA, thereby enabling genome editing by suitable procedures. The only major limiting factor for CRISPR-Cas9 system is the protospatial adjacent motif, which is recognized by Cas9 and is required for DNA cleavage must be located downstream of the guide RNA binding site. So friends, this is all about CRISPR-Cas system. For more updates, please subscribe my YouTube channel Sam Biology Class. Thank you.